Thank you very much for your kind invitations to talk about the clinical challenge for bioprinting. And currently, bioprinting gains more attention for clinical practice due to its ability to overcome the challenge engineerings encountered in tissue engineering for organ and tissue replacement and transplantations, like for a kidney, for a pancreas, for a lung, or a simple tissue like a bone. For example, in case of organ transplantation currently in the US, you can see easily that there is a discrepancy between the number of patients waiting a kidney and the capacity of human organ donors, since you can see that we have a mean of 1,200 days on the waiting list to obtain a kidney for patients on dialysis. More important, it's important to consider that per day, we have a risk of 12 people die on the waiting list for a kidney. Therefore, you can estimate that we can lose more than 4,000 people and patients on the waiting list for a kidney per year. It's a very important clinical need to replace organ transplantations due to the limitations of human organ donations. But not only for organs, it's an important market for, in case of cell therapy, gene therapy, tissue engineering, essentially nowadays mainly supported by cells and tissue banking in US and also in Europe, but also for therapeutics company and biotechs, such as reported previously on the previous report by, for example, bone therapeutics, in case of musculoskeletal regenerations. And you also can see that currently, the most important challenge in view to replace, it's not only for organ transplantation, but also for musculoskeletal replacement like bone, cartilage, tendon, but also for skin transplantations. You can easily show that we have only minor focusing works on organ replacement in contrast for musculoskeletal and skin wound healing challenges for business markets for biotech companies. But however, when you want to replace a tissue or organ, it's important to understand that we must maintain a balance between the capacity of the bioimplants in view to remodeling in vivo, the functionality, the biocompatibility, and the physiology, the anatomy, and the cellular engraftment found in the host tissue or organ that you want to replace. The human body is a very complex machine, and therefore it's important to keep in mind that you maintain all elements on one train in view to have a maximum of chance to replace your tissue like you want. Human clinical application of 3D bioprinting remains a major challenge since it's a large scope of discipline implying the 3D printing. In contrast to non-biological printing, biological printings encountered a major part of discipline from imaging up to the clinical applications. The first important requirement in view to synthesize a complex or heterogeneous functional tissue or organ is to know exactly the combinations and the organizations of the host tissue or the organ that you want to replace. Firstly, the first development is certainly the step one is the imagings. And after that, to go to the applications and the clinical application, you can choose your design approach. Like in bioprinting, you have three main approaches. You have the biomimicry, just to synthesize exactly the structure that you want for a vasculature replacement, for example. You have the autonomous self-assembly, corresponding to the embryogenesis, like a guide to obtain in vivo your desired tissue or organ. And finally, you have the mini tissue building blocks by additions of small units in view to constitute your required tissue or organ. Secondly, you have the scaffold levels. As previously mentioned, is the key questions in view to obtain your desired viable organ or tissue. And for example, in the past, most of synthetic materials have been used. Currently, there is a new development of decellularized tissue from extracellular matrix from human body, for example, and some success was obtained for cartilage, for heart tissue replacement using a muscular decellularized tissue from a human body, and you have also adipose tissue, for example, in breast replacement and regenerations. The selected materials must include some properties implied for the replacement. The printability, to obtain your desired structure, the biocompatibility for in vivo applications and to avoid any toxicity for your cells, vehicles by the biomaterials, your degradation kinetics, but also the structural and the mechanical properties, for example, in case of cartilage or bone tissue replacement. And finally, also the materials mimicry. It's a very important field in view to replace. Sometimes a lot of people forget this materials mimicry, for example, in case of bones tissue engineering, because it seems to be easy, but when you see the strength, capacity to, for the resistance of bones, you can imagine it's not so easy to replace by synthetic materials. The cellular levels is very complicated currently because it's a small unit that you can find in the human bodies. It's important if you want to use bioprinting for transplantations in a human body is to have a well-characterized and a knowledge of the human source of human cells used for the bioprintings. Autologous, allogenic, and something like that is important in view to avoid any immunological reaction, for example. It's also a combination of different type of cells phenotypes. It's not only one. If you use for bone tissue, sometimes a lot of people think that you need only osteoblasts, but no, there is other categories of cells implied in the tissue and the bone tissue remodeling. You need also to have a great understanding of the capacity of proliferation and the differentiation capacity, not only in vitro when you synthesize your 3D structures, but also in vivo in view to obtain a stable and a long-term remodeling to replace the host tissue and organs. The bioprinting technology also is an important aspect in view to select and in view to have a potential of commercializations for the future. You have three main techniques. The inkjet techniques, also called drop-on-demand printings. It's very low-cost bioprinting, but you have a capacity of density relatively low, and also you can maintain a higher viability of your cells vehicles for this bioprintings with a preparation of time which remains relatively low. If you increase the technology by micro extrusion techniques or laser assisted techniques, you reduce the capacity for the second one in terms of cells that could be injected by the bioprintings, and the cost is more important. But for the laser techniques, you can increase the number of cells for example, to replace and to synthesize a kidney for organ replacement, but the cost is higher, and therefore in the balance to be used for a clinical practice and to overcome the organ transplantation, this remains certainly more limited with a long time course for biotech company. Also, if you want to replace a vascularized bones or a vascularized organs, it's important to keep in mind the vascularizations in vitro and in vivo to prepare a maximum of chance to have a cellular viability after transplantations. And it's important to keep in mind that we need to have a well-developed vascular tree in view to have connections and surgical applications for transplantations. And also another aspect, a lot of time and often forget it, is the innovations, because of most of the organs needs to have an innovation in view to functions, like if you want to replace a metabolic functions, like for a pancreas transplantations for insulin release in view to control the diabetes, you need to have an innovation control, and it's often forget. The advantage of the bioprintings, you can perform an in vitro maturation in view to maximize your function after in vivo transplantations. And there is some example for the cartilage replacement. You can develop some force and strength in view to obtain your desired tissue to have a maximum of chance of replacement in vivo. And also, we have also the bioreactor technology for cultivation in vitro in view to develop, for example, a trachea replacement for preclinical models. Currently, there is not a lot of big literature. There is a lot of number of review, but there is not a lot of new paper, original paper on the bioimprinting. There is some proof of concept in vitro to replace a two-dimensional tissue like skin and cartilage, certainly the most rapid access for clinical application. Secondly, for hollow fibers like valve or like trachea replacement, it remains more limited. There is only in vitro testing, not only in vivo testing. And we have more complex the solid organs to replace. As previously mentioned, a kidney, there is only a principle and a concept that there is no evidence of in vitro and in vivo functions of this type of organ. I think it's important to keep in mind that we are only in the first step in the two-dimensional structure for bioprinting. Now, however, there is, as I previously mentioned, different steps to be included in view to mimic tissue replacements. Some combinations of techniques for bioprinting of cell type, selected growth factors, and materials exist now to replace some structures like a heart valve, like myocardial tissue, as also bone tissue and nerve. Therefore, it's already demonstrated that you can find the adapted combinations in view to arrive to your final tissue as desired. But however, it's important to keep in mind that now, as mentioned, at short term, we have a capacity to replace a simple tissue like a skin, hollow tubes or hollow organs like vascular, like tracheal. You have certainly up to 10 years to arrive to clinical applications and to demonstrate the proof of concept in the phase 1 clinical study. And for solid organs, in view to translate in clinical practice, I think that between 10 and 20 years remains relatively reasonable to obtain a clinical proof of concept. But however, we have some positive aspect. We have support of government. We have certainly a capacity of a low-cost productions of tissue, and we have also a large scale of applications. It's a positive aspect to develop bioprinting, not only for university clinical hospital, but also for industry. The negative aspect is the regulations between different countries, different continents. It's also the non-profitable aspect for a biotech company in the first stage of implications for clinical practice, and also to select biomaterials which remains compatible for GMP facilitations and productions. Because for me, the two main limitations in view to apply the 3D printing to replace an organ and tissue is certainly the regulation, as previously mentioned, is you use the ATMP, because most of the bioprinting technologies are essentially focused on differentiated cells. Therefore, it corresponds to an ATMP regulations. And secondly, another aspect relatively underestimated is the ethical aspect, because if you want to replace a tissue and you have a dedicated price for your organ or your tissue, it's a commercialization in contrast to organ and tissue donations that you can find nowadays in our health systems. Thank you very much for your attention.